recording. Okay, so, well, I'm very glad here to present today to Rebecca Hilgenhof. Rebecca, that she's here, she has developed a flourishing career in botanical garden horticulture. She obtained a diploma in horticulture at the Royal Botanic Garden of Kew, and she had several positions as collection horticulturist in the tropical nursery of Kew. So she has worked on the temperate conservation collections, also in the propagation of the plant material required for the restoration of the temperate house, and as tropical nursery supervisor managing the orchid unit. In 2018, she decided to leave her role in Kew in order to pursue a career in plant science, and she joined the Masters in Biodiversity and Taxonomy of Plants at the University of Edinburgh. For her master's thesis, she started working on Solanum under the supervision of Tina Sarkinen and Sandina, focusing on the identification of morphological traits that define the species and measure clades of Solanum. Upon a successful master's, Rebecca continued working on Solanum with a Davis Fellowship at the Royal Botanic Garden of Edinburgh, and this grant gave her the opportunity to continue her work on this giant genus to construct a multi-success online quiz that, well, she will present today. Rebecca is particularly interested in taxonomy, classification, plant morphology, phylogenetics, and also evolution, which she hopes to further develop with a PhD that she's currently looking for. So heads up, everyone, that, yeah, for opportunities for her too. Well, now I leave Rebecca for her presentation. I will ask you, all of you, to turn off all your microphones and videos for Rebecca's presentation.
Okay, I hope that everyone can see my screen now. Please let me know if not, but we're gonna go.
Now we can see the others, the passiflora. Oh, now we can see the screen. Yes, perfect.
It is just a slide, is it? Yeah, Rosie?
Yes, that's perfect.
Okay. Okay, brilliant. Okay, well, hello everyone, and welcome, and thank you very much for joining me here today. So over the next 40 minutes, I'll be talking to you about my work on morphological trait evolution in the megadiverse genus Solanum, which was conducted in partial fulfillment for the MSc in the Biodiversity and Taxonomy of Plants at the Royal Botanic Gardens, Edinburgh, and the University of Edinburgh. Furthermore, I'll be talking about my work over the past year as the Davis Fellow, a position funded by the Civil Trust. This work focused on the construction of an online multi-access key to all groups of Solanum and one for 342 non-spiny taxa. These keys will finally enable us to tame the monster. But before we dive into the two main subjects of today's presentation, I would like to use the opportunity to just briefly start right at the beginning. So as this will enable us to understand how this all came together. So when I was young, I sometimes felt I was growing up in the botanic garden, always in the company of many exotic plants. So it was inevitable for me, once leaving school, to pursue a career focusing on the plant kingdom. My professional career in botanic garden horticulture led me to study at RBGQ for the Q Diploma in Horticulture. I was only meant to stay for the 3-year duration of the course, but as it is so often in life, when you're having too much fun, I ended up staying 3 times longer than initially intended. By the end, I was working, as Rosia said, as the role of the tropical nursery supervisor before my career took a different turn. So during my time in Kew, I not only developed an ever-increasing fascination for plants as a result of the often very intimate relationships that one develops when caring for these, but it also allowed me, through close observations, to get my eye into plant morphology. It was there and then that, for rather obvious morphological reasons, I developed a passion, and I'm choosing my words carefully here, for a particular group of plants. The passion flowers of the genus Passiflora. I was intrigued by the bizarre floral morphology and but I also became interested in other aspects on their cultivation, such as taxonomy and classification. As a result of the endeavor to comprehend this wonderful group of plants in its entire, I decided a slight change of career was necessary. With the purpose of becoming botanically trained, I left my position in the horticulture department at Kew in order to pursue the master's. This taught postgraduate program included 9 months of both lectures, with an additional 3 months dedicated to our MSc thesis. So when the list of available summer project choices were released, a particular one straight away caught my eye, with the title Characters That Shape Evolution, Identifying Morphological Traits That Define the Species and Major Clades in Solanum. For me, that was a very easy choice to make, as it included one of my favorite subjects, plant morphology, hereby focusing on a group of plants that previously come across during my time at Kew and doing some of the fieldwork I undertook in the neotropics. Initially though, when I was thinking about morphology in Solanum, I personally thought there would be only little morphological diversity, as all Solanums have a specific floral Bauplan, as here illustrated by this Chilean potato vine. They possess 5 merous flowers, fused corolla and calyx, stellate to pentagonal corollas, short filaments on the stamens, and anthers opening bi-terminal pores. But, and it's a big but, shortly after starting my thesis, I had to realize how wrong I had been. For example, there are species that possess campanulate flowers, such as this one here, although most have stellate or pentagonal corollas. They also possess a variety of different corolla colors, as nicely illustrated here in the slides. But also stamen morphology is diverse, for example. Stamens can be either arranged in tight cones or completely free and spreading. Their shape can be varied too. The anthers of some species are fat and stubby, whereas others are long and thin. Even cordate anthers can be found among this group. Stamen dimorphism too occurs readily in Solanum. A good example for this is the Solanum rostratum here. So this morphological diversity is likely to be linked to buzz-pollination syndrome. All Solanum species are buzz-pollinated. They rely on bees that are capable of buzzing to access the pollen locked inside the porocidally dehiscent anthers. Hereby, a particular sonication frequency is required to eject the pollen. So this frequency required, however, varies and is dependent on the size and the size of the anther in relationship to the free portion of the filament. This means that only the perfect bee match will be capable of producing the right buzz in order to access the highly nutritious reward. Before I will present my findings from my MSc thesis, I briefly wanted to provide an overview of the genus Solanum. Solanum is one of the world's megadiverse angiosperm genera, currently containing 1,242 accepted species, which is nearly half of the species contained in the entire family Solanaceae. It contains several economically important crops, such as the potato, tomato, and the aubergine. The genus can be divided into 10 major and 49 minor clades, as here highlighted on the right, on the phylogeny published by Salkinen et al. In 2013. And also, as we've just seen in the previous slide, they are morphically very diverse in respect to reproductive, but also vegetative traits. So thank you to the tremendous work efforts and dedication of Solanum experts worldwide in the past and of course in the present. Clade level revisions exist for 31 minor clades and is ongoing for an additional 4, but unfortunately is still missing for 14 minor clades. As a result, there's a lot of information available in published format, which is extremely useful for recording the morphology. On top of the published work, we do have great taxonomically verified specimen database that has been built over the years and is being kept up by Sandy. The database now includes over 100,000 verified collection events from over 450 herbaria. In fact, the data is now so vast that when plotting these botanical records, they provide an outline to all the world's major continents as illustrated here in the slide. Members of the genus Solanum are widely distributed, whereby they occur across all temperate and tropical continents. As highlighted by the density of collection visualized in the slide, the highest number of species is found in tropical South America, where they occur in an incredibly wide range of habitats, from the tops of the Andean mountains to lowland rainforests and dry forests, as well as desert and wetland ecosystems. But not only Solanum follow this trend, but so do our collaborators shown here in the slide. These experts are spread across the world and have been extremely helpful and fundamental part of my work. Thanks to all of you, I've been able to do this. So I started a 3-month MSc project, and this talk is based on some of the results, as I mentioned earlier. So I will present today the following 3 major points. I'll provide an overview of morphological characters I recorded across Solanum clades. I will touch upon morphological trait evolution, so the mapping of these characters across the phylogeny of non-spiny species to study their morphological trait evolution. And last but not least, I will present the identification key and how we use the characters to build the interactive motif access key for both the Solanum groups and the species of non-spiny members of Clade I. So my thesis set out to explore the origin and evolution of 32 taxonomically important vegetative and reproductive morphological traits found across Solanum. These 32 characters represent 13 vegetative, as some being illustrated here on the bottom left, 14 floral characters, with a few being here on the top, and 5 fruit characters here on the bottom right. All characters were chosen based on expert knowledge and those used in clade and species descriptions of published monographs. So if you have a close look at the vegetative and reproductive traits, here are the 13 vegetative traits. To the left, there are 3 examples of that I've chosen that I wanted to touch upon in a bit more detail. For example, here on the top, number 1 is growth forms, where we identified the following stage, which includes epiphyte, herb, herbaceous vine, woody vines, shrubs, and trees. Then number 4, we've looked at the different hair types that can be found across Solanum, including those highlighted in the center column here. And then number 9, we also looked at leaf types and identified a number of traits as illustrated here, ranging from entire up to what one may consider as being compound. Here are the floral traits that we assessed, 14 in total. This includes, for example, the corolla shape, here on the top, number 5, including stellate, rotate, campanulate, and oscillate. Then we've also assessed Corolla Color, here by identifying 4 states, including green, white, yellow, and purple. And another example I wanted to highlight are anther appendages. Although these can be absent, and the appendages can be apical, as here in the picture, in the yellow picture, then anthers, anther can possess anther horns, or they may possess anther beaks. And here are the 5 fruit traits. These, for example, include the type of fruit, which includes fleshy berries, leathery berries, hard berries, and dry berries. But we also looked in fruit color, whereby we've identified 4 states, including white, green, yellow, orange, red, brown, blue, and purple. However, such work does not come without its challenges, in particular when looking at morphology. These occur quite rarely. For example, we know that tubers evolved multiple times in Solanum, but the question remained if all of these tubers are homologous in these groups, both anatomically and genetically. Similarly for fruit, in Solanum, there's still very little known about the pigments or pigment ratios present. Neither do we know about which genes are involved. We know, for example, that the carotenoid pathway results in shades of orange, yellow, and red. Red, however, could also be the result of anthocyanins, for which reason this character was assessed with caution. Other examples for fruit colors that were difficult to interpret were blue, such as this one here, as well as brown, for which reason these were kept separately from the rest. On the other hand, we also had to assess continuous characters such as leaf type, which was challenging to score too. These had to be categorized, and in the case of leaf type, this was done by quantifying the depth of the sinus to the distance of the midrib. For example, in lobed leaves here, the sinuses reach from a quarter to half of the length to the midrib. Here in the next slide, based on the findings of Sakinen in 2013, I wanted to just touch upon the structure that we find based on molecular analysis of this genus. For those that are less familiar with what species or clades are included in Solanum, I just wanted to highlight those that have put pictures on here. So for example, we have got the tomato and its crop-right relatives in the tomato clade, which is sister to the potato and its vast number of crop-right relatives in Pitota. But we have the aubergine, which is part of the eggplant clade in the non-spiny solanums. But we also find other examples like the well-known kangaroo apple here in Arceselanum, the pepino, which is part of Basathrum, which is over here. Then we have got Solanum dulcamara in the dulcamaroid clade. And then last but not least, I just wanted to point out this tree tomato, which is part of the Cyphomandra clade. This phylogeny from 2013 had about 33% of species sampling. However, since then, many more sequences had been published in GenBank, and many more unpublished sequence data was available for our collaborators, resulting in a need for a new up-to-date supermatrix, which we would be able to use for the reconstruction of the ancestor traits. So in the Sakinen lab, we constructed such an up-to-date Solanum supermatrix based on Bayesian inference and maximum likelihood analysis. The final supermatrix included 743 taxa, which equaled 60% of all Solanum species, hereby presenting all previously identified major and minor clades. The phylogeny was constructed utilizing 2 nuclei and 7 plastid markers, covering a total of over 10,000 base pairs. We recovered with moderate to strong support all previously recognized clades, and to my personal excitement, revealed novel relationships as the result of the increased species sampling. So we recovered 4 new clades, or 4 new groups, namely the Valdiviense clade, including 3 monotypic lineages, Anomalum, Polygonum, and Eucanthum, as well as confirming Leandro Giacomi's findings of the Trichome. Unfortunately, though, our supermatrix shows only weak backbone support, which did not allow us to draw well-informed conclusions about the evolutionary relationships between the minor clades. Resulting from the research led by Edin Gagnon, currently in preparation, which in addition to the supermatrix analyzed plastid and target captured datasets, we understand that there are hard polytomies, and these are presently unresolvable and may be never fully resolved. However, I will stop here and like to refer you to a very comprehensive lecture on this topic presented by Adeline 2 weeks ago, which is available online on the Solanacea Seminar Online YouTube channel. So for the ancestral state reconstruction in Clade I was chosen as it presents one of the best sampled and taxonomically well understood clades. Supermatrix included 206 taxa, which equaled 60% of the 339 species present in Solanum Clade I. In order to best illustrate our findings, we transformed this part of the phylogeny to be displayed in the same manner as here on the right. Overall, we had observed high transition rates in 6 reproductive and 2 vegetative traits. I will be showcasing a few of the most interesting findings in the next few slides. Anther appendages are found in 3 groups, including apical anther appendages and anther horns, but also anther beaks. However, anomalostemum is not presented here and is not part of this analysis because it's part of the spiny clade, despite not possessing any spines. Our results have shown that most of the species in Solanum Clade I do lack anther appendages or horns, with anther appendages only found in 4% of species and anther horns in only 1%. Not at all surprising, the absence of anther appendages was found to be ancestral in Clade I, and in Clade I, hereby the transition from the lack of anther ornamentation to the presence of anther appendages was found to be slightly more common. Here I'm showcasing a character, previously I've showcased a character that showed a low transition rate because it's preserved, the characters are well preserved in certain clades. But now moving on to those characters that show high numbers of changes. So most solanums in clade, one example for this is the filament-anther length ratio. Most species of Solanum Clade I had a small filament ratio, i.e. stamens with clearly shorter filaments than anthers. Only 29 species had a medium anther filament ratio, and only 4 species had a filament ratio where the stamens had filaments longer than the anthers. The highest transition rate was found from small to medium filament anther ratios. Hereby, short filament anther ratios were found to be ancestral in Clade I. If you have a look, for example, at the Morphological clade, which is one of our best samples in the Clade I phylogeny, we can see that between sister species, we have changes between a short and an equal filament-anther ratio, which quite nicely highlights that closely related species might have a different pollinator. Our second character showing high numbers of changes is Corolla Color. 4 main color types were identified: green, white, yellow, and purple. The most common Corolla color found in Solanum Clade I was purple, followed by white, yellow, and green. The highest number of changes was found from purple to white and from white to purple, with purple being ancestral in Solanum Clade I. Again, hereby we often see changes between sister species, where they change from white to purple and purple to white. However, there are also examples, like the tomato clade here, where yellow is the most predominant color. Fruit color. We found 8 colors, fruit colors present in species of Solanum Clade I, with the most common fruit color, to my surprise, being green. This was followed by black, orange, and yellow. The highest number of changes were found from green to colored fruits, from green to black, or green to purple here, from green to red, from green to yellow, and from green to orange. It is interesting that changes from green to red were more common than changes from red to orange, and vice versa. Although this probably here is to be an indication of the current pathways being present. In terms of vegetative characters, we looked at hair types. We were able to identify 4 hair types that are present in species of Clade I: simple eglandular, simple glandular, bayonet, and dendritic trichomes. Simple eglandular trichomes was the most common trichome type found within Solanum Clade I, followed by simple glandular trichomes, bayonets, and dendritic trichomes. The highest number of changes was observed from simple to simple eglandular, followed by simple eglandular to dendritic. Simple glandular trichomes were found to be ancestral in clade I, with all other types of trichomes derived from these. So this was a quick overview of the major findings from my master's thesis, whereby we can confirm that truly it is all about the buzz, as the highest transition rates were found in traits that are related to pollination. However, here in the next slide, I also wanted to show a few things that we were able to identify along the way, such as certain synapomorphies found amongst the clades of Solanum. So the most obvious synapomorphy, and that has already been previously identified, is that when one looks at armed versus unarmed species, is that we can actually separate the genus into half, with half of the species possessing spines or prickles, and the other half are complete, the other half lacking the prickles entirely. Similarly, such clade synapomorphies can be seen in other clades. For example, we have the anther horns here in Romania clade, the apical anther appendages in tomato clade, and the enlarged anther connectors of Pachyphylla. Interesting enough, these three groups were previously by some taxonomists separated based on these morphological features. So synapomorphies do really exist in other clades of Solanum, and I just wanted to draw out a few other examples. For example, the cup-shaped pedicel insertion of the Dulcamoroid clade, the single pseudoscipio found in Anaricomemon, the cordate anthers with the apical beak in Anomalostemon, the geniculate hairs found in Inonatum ongonata trichome clade, and stellic hairs on the outside of the anther cone found in Crinitum. But on the other hand, we can also observe certain traits, such as tubers that evolve 4 times within Solanum, twice here in the spiny clade, and also twice in the non-spiny clade, of course, including the crop bright relatives of the potato here, part of the potato clade. Well, what we also found that there were certain cases where we were not able to identify a single trait that would enable us to distinguish a particular clade. In those cases, it is a combination of traits that allows the identification of these. For example, on this slide here, we do have a paired or geminate leaf, a shrubby habit, and a white corollas, short stubby anthers, and green fruits, which is a good indication for it to be a member of the geminator clade. With this in mind, and the data I had available through the analysis, we decided to construct an online identification key for all minor clades of Solanum and the 342 species of non-spiny Solanums. For the purpose of the key, the data set was revised and expanded to 41 characters to include other important aspects to aid identification, such as geography. For the construction of the online key, we decided to use the software Expert 3, which is hosted and maintained by the Natural History Museum in Paris. Expert in general is a what is Expert? Expert in general is a taxonomic management system for the storage, edition, and online distribution of descriptions. But more importantly for us, it allows the creation of multi-access online keys, in addition to the interactive identification of specimens. But why did we decide to use Expert for the construction of the key? Well, first of all, it is completely free. It is available online, or in the case of Expert 2, also locally. It is very user-friendly and does not require any special computing skills. The user can choose the characters to be observed, identification if possible, even if characters are lacking. The characters can be used in any order. The software also includes the possibility to print a single access key for field identification if needed. The key can be completed with illustrations such as pictures and drawings and text explaining the terminology used. Correct identification can be obtained despite errors made by the user. And the characters are weighted according to the skills and the abilities of the audience. So in order to assist any first-time user of the Expert key, we put together this quick guide to Solanum online keys, which I just want to explain to you briefly. So everyone that will access, be able to access the key via the hyperlinks on the Solanacea source website will be able to use this format. So it highlights that for Expert, and one particular learning curve for us was actually using this key, that polymorphism works much better for identification, simply by the fact that interpretation of certain states might be difficult, and it also depends on the eye of the observer. So in case of uncertainty, for example, multiple characters or multiple options should be chosen per character in order to not eliminate the species that could be the result. It should only be using characters one sees. We can search the characters here as highlighted. There's a history tab which allows us to track back which characters we have already used. We can make choices with the submit button here, and our remaining results will be displayed on the right hand side panel. We've also put in test specimens so every user can practice prior to using the key in the real world. What we also illustrate here are characters in Solanum that are most informative when it comes to the identification of species as a result of prior knowledge, but particularly through the key phase testing that we have been doing over the past few weeks. So these characters, these major characters are shown here on the left-hand side. Particularly geography is a very good character to use because it's most likely that people know the location of the species. However, also other characters that are very informative are prickles, but obviously this will only be applicable for the clade level key, as the species level key only deals with species that don't possess any prickles at all. We also we identified fruit color. Obviously that depends on the presence of fruit, but these are very obvious to the observer's eye. The same goes for Corolla coloration here, but also we try to use informative vegetative characters, as these should be present throughout most of the season. One example here being the absence or presence of pseudostipules, either a single pseudostipule or a paired pseudostipule. However, before I'm getting too sidetracked, to just show you what I've been putting together on the slide, I think the nicest thing to really illustrate you what the key looks like is to actually go online with you guys, and I hope my internet will be okay. So what I will be doing is to just give you an example, randomly chosen from me on this species level key here. And then if you like to, I will also go into the key to the Solanum groups. And if anyone in the audience is interested to throw some characters at me, and we will see which clade will be the result of that. So one of the most obvious characters, as I said, is geography. So if we click on geography, particularly obviously in geography, I should probably mention that we're using, that we're following the database of the International Working Group on Taxonomic Databases for Plant Science in order to allow consistency with the evaluation of the geographic location. So essentially the user, and as I previously mentioned, there will be pictures, but also descriptions. Here in that example, we're dealing with Northern Europe, which for example includes the UK and Finland. However, I'm more of a tropical person. So personally, I'm going to choose a species from Western South America. So once the user has chosen Western South America, which is highlighted here, one needs to submit it. And all of you already seen that among the 40 taxa, we now have 190 taxa that are recorded to occur in Western South America. Hereby, it will be the most common characters. For example, through color. If you go to color, all our characters are illustrated, and so are dates. Say, for example, a yellow fruit, I would again submit this. So that means now 20 taxa are remaining recorded to occur in Western South America that have yellow fruits. And these are listed to the right. Due to the unstable internet I'm currently having, it's going to take a while until the images are loading. Here it is. As I'm probably not going to have any flowers present, what I will use now is leaf, and I've got compound leaves that toothed. It's already telling me that there's only result. And present, so here we go. One remaining taxon and Solanum leucopersicum.
Rebecca,
if you go,
oh, sorry
yeah.
to interrupt you. I think that your screen, oh, it's frozen, but now it's working. Yes, now it's not working.
Okay, thank you, Ratia. So user can then go into the result, and unfortunately, everything is loading currently, but here we go, slowly but coming. We put additional notes here on the top in order to get the available information on solanaceae, the link to the species description. This will present here in sense of characters and used in order to construct a key. And unfortunately, it's not loading for most of the species, but also for all the clades. We've included also the user has a visual confirmation if the task was keyed out is coherent with the result. Here we go, and we've got a tomato. Isn't that nice? Right. Now that was a quick one. So if anyone would like to join me, I know if no one puts their hand up, Tina's probably gonna help me. So please, if someone like to throw some characters on me, we can have a look at the clade level key as well.
If you want to do it in the chat box, I'm also observing that, so I can say if I see something there. You can start with geography.
Yeah, if you can just tell me, because it's I find it a bit confusing.
Should we do Australia for the fans of Australia in the audience?
All right, Chris Martin, this is not, that obviously is for you. Let's
you can also
find
search and it will take you to Australia.
Search. Yes.
Just find and search, I think, on the page rather than, but try it. It won't find because it's not the state. You can look for character there. But on the page, if you go
yes.
find and search on your keyboard. Oh, Control F.
Yeah.
Oh, you have a Mac. I don't know how it's
that's a Mac. Exactly.
Apple F or Mac.
Apple F.
I'm using a different. All right. Sorry, everyone. Oh, here we go. Australia. It's now highlighting it here. I'm not sure that's the best way. If you don't mind, I'm just gonna look out Australia. Here we go. There was New Zealand, so it can't be too far. Here we go. We've got 19. So we submit that.
Oh, we on the group level key, right? Good.
Yes, we are on the clade level key, group level key. So what does your specimen have, Tina?
I feel red fruits.
Okay, you can feel red fruits, that's good. Here we go. We highlight orange to red there. Submit.
Also in this key, actually, we've lumped orange to red because people see them differently. We realize that otherwise it's impossible in the key.
All right, so there's your remaining options.
Okay, I feel spines. I have spines.
Spines, okay. Here we go. Present, submit. Down to 15, down to 8 groups.
And now it's the question of what Corolla color. What do people feel?
Hang on.
I wonder whether there's a choice of yellow. Is it left? Is there anything that has red fruits and yellow flowers in Australia?
Leandro says white.
White. Oh, Leandro is feeling white.
Okay. Unfortunately, I don't can get Corolla color. Can Leandro choose fruits?
You can search for Corolla color, but you might not be basically separating. So in the because sometimes I don't actually understand why. Leandro might know the answer because he uses these keys a lot. Why some of them disappear at some point? Maybe because they have no distinguishing power.
That could be. So
but there
it was
you
white.
See, there's
Leandro
one
said,
yellow.
wasn't it?
Yeah.
There's one yellow. Which one would
you
choose? Yellow or white? How about
I would want to know what that yellow is, and Leandro does the next one. Yeah.
Okay. Let's do the yellow one. And there we are in Arcantophora.
Ta-da! Okay, Leandro, you take the white one. So sometimes that search of the character is really important because they disappear from the list for some reason. I think it's a bug in the software.
Here we go. There's our characters. Let's take that one out. Here we go. So you can alter the history. So you said white Corollas, right? Here we go. Here you go. There is 8. Submit. Tina, any other character?
You can try the white, and then we can take more characters once we have 8 left. Okay. Fruit and calyx. I think, Rocio, what kind of calyx do you see?
Oh, I would like to see an inflated one there.
Yeah, let's go for it.
Okay, right. Here you go.
So we've, in this key actually, Rocio, we've started changing the states just because as a non-expert user, we felt like people got in the testing stage a lot of things wrong and right. So you can go back and see our states that remain, but please give feedback. They're very easy to change. It does take time, not undermining the amount of work Rebecca has done, but what I mean is they are possible to change. So email us with feedback.
All right. Anyone next character? Because we've got still 5 remaining groups.
Okay, here we have a question also. If you're taking questions already about
Yeah.
limbos. She said that she sees Solanum vetaceum comes out being in Australia, but it's not native there. So the cultivated adventitious introduced ranges are included in geography.
Yes, we've had to really soul search on this one. I'm answering because I've been responsible for the recent change since Rebecca has started working more on the analytics side. So we lumped those two, unless it's an obviously cultivated species. In cases like tomato, where you find lots of how do you call these ones that have escaped specimen populations. So then, because the user will not know which one it is, they're coming across a weed, which most Solanum species are anyway. The likelihood of error is big. So we've included the really common weeds that are not native. And although Betaseum isn't...
No.
Wait, go on. Oh, so although Solanum Betaseum isn't super weedy, we've just put it in because it is quite commonly cultivated in areas that we might as well put it in there.
Okay, good. Yeah. Novilin wants to ask something more about that? Or... Okay, well, we can continue trying to, yes, with those 5 groups, the remaining taxa.
Well, Rosia, do you want to choose another character?
Let's see, how about fruit color?
Fruit color, okay. Well, I think we already had, because that's not an available option.
Okay, so which other we can choose?
Maybe prickle type?
If they're hairy fruits.
Yeah, hairy fruits.
Okay. So they are present. Here we go. Here we go. 2 choices.
And actually, that points into just one, because we've split the old world group into these subgroups. So what it's saying is that you're in the old world group, and within that, you're likely in the dioicum group. However, we have not put every single subgroup of the old world group in because they are not perfectly grouped, which is a bit dodgy.
Anyway, so as we are probably very likely, and to Chris's enjoyment going into dioicum group. So again, similarly to what we would have seen to the species level key, the user will be presented with just a selection, because obviously sometimes clades are rather large, just a selection of what species within it look like, both for reproductive as well as vegetative characters. And again, a bit more information here and the consensus. So now I'm just going to go to where this information will be available, and also highlight the fantastic work that Sandy and Tina have done recently on the Solanaceous source page. So we do have our own tab here for the identification keys. Of course, it names everyone involved, which is a lot of people because it's a rather large project. It will provide the keys for both the species level one as well as the groups, with a bit more guidance and information as well. And if one looks further down, also what it will provide is links to already published keys, dichotomicities to certain groups. These are all alphabetically ordered. But just go down right to the base, down to Vinlandii. And I think everyone, or a lot of people in the audience will probably recognize their own work being displayed here. Here we go. One other aspect as well that I like to highlight is for the species. So as I said, or as I showed within the species level key, we have put a link. Unfortunately, it's not hyperlink. We put a link in there that allows the user to be directed right to the species page on the Solanaceae source website. And here we get the overview, much more elaborate species description, as well as additional information.
And this page
also
includes notes about how to separate species from their most likely morphological beasts. So that is the kind of missing link that's not available in the key, but you can find it in the Solanaceae source.
And then also the images that will be, or we have used to put the plates together, they will be here available individually and also downloadable under the CC copyright. The nice thing, like with everything on Solanaceae source, it's really accessible, and so are our keys. Anyways, let me just get back out of this one here and just go back to this one here, because I just want to end my talk with this particular slide to say a thank you to everyone involved. It's a large project, so we could have not done it without everyone, and also obviously people from the present and their fantastic work on the genus Solanum. Well, thank you very much for listening, and are there any more questions?
Thank you very much, Rebecca. It's a real great presentation and a huge amount of work. It's amazing, this key. Yes, it's kind of a treasure to identify Solanum. So we are open for questions. Yes, if you want to turn on your microphone, ask directly, or just ask the questions on the chat. Yes, of course. A lot of people very excited to use the key now. So nice.
Yeah, we just maybe stop sharing for the. Here you go.
May I ask a question?
Sure. Yes.
I just wanted to know concerning the key. So if I remember well, you have a gradient of color from white to green, and this gradient of color is also a gradient of color that you can see for the characters. And when it goes to green, it means that the character is very discriminating. That's it. And when it's white, that you have less discrimination if you choose a state for this character. Am I right? Or I was just wondering that. I was not sure about the signification of this gradient of color when you have the list of the characters. You know?
Yeah, I know what you're kind of meaning. So when you're in the key,
yep.
Do you mean the gradient of color in the bar on the right hand side where you see the results?
Not on the right when you see the result, but on the left when you choose the character. Of course, on the right also you have the possibility that your choice is very strong or not. I don't.
Do you mean the one bar at the bottom that has different shades of green?
At the bottom, but also the character themselves. If you look, the characters are either surrounded by green or surrounded by white. So I was wondering if this difference in color is that the software is making you, is telling you this character is in preference, you choose, if you want to really discriminate between species and group of species, you should choose this character. So I just wanted to know if this color was a sign of better discriminative power.
I mean, in a way, not, because I don't know how it's doing it personally, but what I would answer is there are characters that I would call these winner characters, like key to happiness, highway to happiness characters that you see cup-shaped pedicels. If they're obvious, bam, you're in Dulcamoroids, right? So it's a highway to happiness character. One character, you're in it. But in most likelihood, if you think about species diversity, so grab a random solanum out of a bag across the world, you're unlikely to have a dual choroid.
Yep.
So in other words, the discrimination power is massive, because when you have it, it gives you this highway to happiness. But how often are you going to have it? So what that key does is it doesn't highlight these highway to happiness characters. They are white. What it does highlight is things that split the group into halves.
Yeah, like geographic stuff or spines or things like that.
Yeah, spine is classic.
Okay, good.
However, it depends. Individuals are quite different which one they like, dropping these odd balls off, or keying them first, or like looking, oh, do I have these highway to happiness characters in my specimens? Let's look. Yep. Oh no, oops, I'm in the bog. Yeah. But what is in our power, and what the software does, is it proposes the order you see the characters in is in our power. So we can say, put this on top, put this on the top of everything. Okay, like good. Geography, we put it on the top. So that's something.
It's you put it. It's not the software itself. It's your choice.
Okay, I know. Guilty.
Oh no, that's fine. I think it's really good, but I just wanted to know if it was the software itself that was doing an algorithm.
But the software does the coloration.
Yeah,
so that's
but the software does the coloration. Okay, good.
Yeah.
Good, thank you.
Really good question.
Thank you, Xavier. Yes, there is also a comment of Franco that regarding the character mapping, that it could be interesting to do this same morphological mapping using anatomy or histology or both. Like, for example, it could be some information on other characters like pollen, seed testa, fruit histology. What do you think about that?
Yes, I think if I might take that answer. I think the nice thing is with Expert that you can upload additional characters, but you can also obviously put in manual characters. So this is only the start of the key. I think the nice thing is, and it's something that Sandy has highlighted in her previous presentation, is that monographs may be stagnant. However, this key, particularly this online key, is something that constantly develops. So it develops on being used, it develops on feedback, it develops by additional character and additional work being put into. And I think the more we can put into, the more accurate we might be by the end of it. So yeah, but then it's nature, isn't it? So, but yeah, the answer is it's pretty great to have more characters.
Yes, great. Well, there are more congratulations. There's also a question about from Federico Roda. I don't know if you want to turn on your microphone, or I can wait for. Okay, so it's about like he's interested in whether there is a correlation between. Oh, he's here, I think.
Yeah, again. Hi. Yeah, I wanted to ask. Great talk. If we are working with trichomes in my lab and relationship to herbivory and secondary metabolism, and I wonder if there is a correlation between the ecology of the plants and the types of trichomes that they have, or if you didn't test for that, if there is data to test for that available right now.
Okay, well, I didn't test for that during my MSc because it was a rather cramped 3 months that I had in order to undertake that project. However, I did refer in the discussion because there are papers out there, and particularly in tomato clades, that deal with herbivory and glandular trichomes in relation to that. So, and I think it's definitely something that should be investigated more in the future.
Okay, thanks.
So just about the collection data, for example, herbarium data could be used to capture the global environmental or climate data or cloud cover. We've started really doing this more and more with Edeline Cagnon, who's in the audience, and she might want to comment, but a cleaning of the herbarium data is always needed. The georeferencing of every single specimen isn't amazing. But with the cleaning tools Edeline has looked into and developed herself in our scripting, you can really clean effectively because we have so much data, never too much, but what I'm saying is that there's enough to throw some out. and you're still left with potentially more than 20 collections per species. So it allows you to get some idea, but they are very macro characters. So I wonder, in terms of herbivory, I would maybe speculate that the data isn't quite there, but you could start looking at something. Is there more herbivory in dry environments, i.e. is it more likely you get glandular hairs in drier environments where there's herbivory? You could do something, some kind of exploratory, and super happy to collaborate, because often our lab just generates very basic data, but we might need bigger brains to keep investigating. So often this is like a roadmap that we hope that people like you would eventually want to use and explore more questions, because once you have a phylogeny, once you know the names are okay, once you know the specimens linked to those names are okay, and you know the pros and cons, the amount of cleaning necessary, you can start doing something. Whereas if you don't have this framework or roadmap, where you're going to go in those bigger questions. So we hope just to do a solid roadmap and then offer collaborations, or once it's published, it'll be there.
Yeah, and I guess in herbaria there is no data about the kind of herbivores that they find in plants. We are collecting that kind of data in our lab, but I guess that's not usually found.
How can I can comment on that. Actually, I don't know if you're familiar with the work of Emily Meinecke. There is trace of, you can quantify herbivory in herbarium specimens. So you can do things like look at the type of damage there is on plants. And I know that you can distinguish it between different insect types and quantify the amount of damage. So there could actually be a lot of that could be an interesting piste d'investigation, like something you could actually look at.
But
So
you
there
can,
might be more there.
The amount of digitized specimens and now the need of these kind of digital projects, maybe in undergrads and whatnot, you really could at least give it a go.
but
We
you
now
can,
know
you can differentiate. Sorry, you can differentiate between what has been the damages by the herbivores before the collection and after the collection, while it was, I don't know, in herbaria. Can you make a difference between the two?
There's a possibility. I'm not entirely sure. I think that I'm not. I think this is the type of project you want to collaborate with entomologists.
Okay.
But they can definitely like look at the type of the damage. They look at it under microscope and they can see if it's like what kind of mouthpieces, is a puncture, or is it different types of nibbling from caterpillars or things like that. So yeah, potentially yes, I would say.
Yes, and actually there are a lot of work that has been done with fossils to see how was the damage on the leaf specimens, fossil specimens, and then to assess how was the populations of different insects according to those plant fossils. So if that can be done with fossils, probably with the specimens, I'm sure that yes, with the living plant specimens for sure. Yeah.
Okay, thanks a lot.
Yeah. Okay, if there is any one last question for Rebecca. So
can I ask one? Yeah.
Sure.
Hi, Rebecca. That's this stuff is so cool. Yeah. I guess I'm interested sort of broadly. So you have this kind of, not everybody has the same perspective right now on all the characters that are sort of occurring in this big genus of beautiful plants. So I guess I want to ask just generally, are there a few things that sort of along the way you've pulled out as like now that are your favorite, like coolest morphological traits that you've seen pop up in certain clades and lineages, right? I mean, what are some of the neatest things that you've seen?
Well, for someone that, as I said, I was quite, how would I term it? Well, before I got into solanums, I wouldn't have known how much morphological diversity there was in the first place. Like I was absolutely mind-boggled once I started working on it. For me, I like oddities when it comes to growth form. That's all my personal thing. So I would say like the climbers, Toroidea, Herbisticum clades. I have to say I favor non-spiny, probably because they have less danger whenever I happen to go out in the field. But I think the climbing ones, they're the ones that particularly stood out for me personally.
Yeah. What a surprise, really.
And Eric did not pay me any money to say that. Rocio, I can't hear you.
Oh, Rocio, you're on mute.
Yeah,
I think Rocio is saying that.
Yeah, yeah.
I was just muted. So yeah, so I was saying that there are some comments about if you use herbarium specimens or to score the glandular trichomes, or how was the scoring of that, or just across the phylogeny?
Rebecca, you're on mute now.
Oh.
It's about miming today. No, so mostly the resources that I've used would be available literature, monographs, obviously Solanaceae source. I think I can't say it often enough without having that online freely available resource that so many people have helped to establish. That's where most information come from. Sometimes in case of uncertainty, if I didn't quite believe, well, that sounds probably a bit strong, but if I wanted to confirm that's what people said was the true story, I would go and look at specimens, particularly when it comes down to trichome types, but more so the more complex branch ones, because that's a completely different story. One looks into this. So.
Nice, yes. And Franco is also commenting if probably for the mapping, it would be interesting to do mating systems. I don't know if you have already tried that.
We do have sexual systems as part of it, right?
Yeah.
Although it will be a character. I think, sorry, I was a bit nervous earlier, so I probably have missed out a few things, but the characters are waiting, so probably one of the reasons that there would have come up because we give a system, not know if you don't read them, that's the reason why it's not one of the first characters to be observed in the key, but it is present.
And then there's anther-dimorphism or stamen dimorphism. So that's again, Rebecca has such a passion for morphology, so she split it into different types of stamen dimorphism. So you can have the filament length varying between stamens, which is one type. You can have the anther being varying in size, or you can have both. So that was really interesting to look at, obviously to do with pollination and kind of mating system as a whole, but different to the dioici and anthromonoici that you recorded as a whole. But again, not all of those are confirmed. So sometimes we suspect something is behaving anthromonoiciously. So we took the ones that are indicated based on morphology as acting anthromonoiciously as anthromonoicious, although obviously we're not claiming we definitely know that is the case. So we had to make these kind of difficult calls that you referred to, Rebecca.
Yeah, well, that's great. So, well, we're finishing now the seminar. If you have further questions, you can always email Tina, Sandy, Rebecca. Well, of course, the next Friday we'll have another seminar, and there is a small change in the presenter. Like next Friday, we'll be presenting Iris Peralta about integrative taxonomy and classification of wild tomatoes and close relatives, insights for conservation and use of genetic resources in plant breeding. So here's Iris, and yes, we will looking forward for your talk. So thank you everyone for coming here, and see you next Friday.
Thank you, Rocio.
Thanks everyone.
Thank you, Rebecca.
Congratulations, Rebecca. Yes.
Bye bye.
Great talk.